Yeah, I'd love to, start by thanking Teresa and the organizers for, inviting me to talk about Biodesix's AI platform as well as our mass spec platform for discovery of immunotherapy tests. Just a little history, a little bit on Biodesix. We are a liquid biopsy based diagnostic solutions company. We span the entire gambit of the discovery pipeline, everywhere from discovery to development to commercialization. We're actually, although I'm talking about proteomics and mass spec today, we're actually a technology agnostic company. We use genomics, radiomics, and proteomics to discover blood-based tests. We focus on tumor and host immune profiling. From there, once we've discovered a classifier, we have 2 highly certified clinical laboratories that take that through, CLIA validation. We have a very established product development pipeline, as well as a strong quality and regulatory department, to take tests through CLIA as well as other certifications. And then we have quite a bit of experience with commercialization of tests. To date, we've successfully commercialized 6 diagnostic tests, and we have 3 additional tests in our pipeline. I have a great deal of experience with reimbursement, and we have maintained an in-house billing and customer care to support all of these tests. Now, our discovery process is really based around our AI platform that we call the Diagnostic Cortex. This is an artificial intelligence platform that's based on deep learning nets. We have multiple layers of abstraction of the data that goes into the Diagnostic Cortex, along with machine learning based methods for error correction, things like preventing overfitting and generalization, as well as boosting data to make it look larger. Now, to feed the Diagnostic Cortex, we use a number of different techniques. For tumor profiling, we use, in biomarkers, we use genomics, both DNA and RNA based sequencing. We use proteomics. It's something as simple as ELISA up to, high density mass spec based, proteomics profiling. We also use radiomics. These technologies are really based around digital droplet PCR, next generation sequencing. We do quite a bit of LCMS in our proteomics as well as ELISA. On the host and immune profiling side, the biomarker classes that we look at are proteomics, post-translational modifications of proteins, as well as genomics. And the technologies here are based on, our proprietary deep Maldi mass spectrometry platform, liquid chromatography mass spec. We've just adopted a new technology from Bruker Daltonics called the PACEF timsTOF mass spec platform. That's a next generation platform that lets us go much deeper into the proteome than we have in the past. Also use ELISA, and then rely heavily on next generation sequencing. Now, all of these different platforms, the technologies that we use, we've optimized over the years to generate really high quality data to go into the Diagnostic Cortex. One of the tenets of our platforms is that we're true believers in garbage in, garbage out. So we pick these platforms so that they're extremely sensitive. They produce, at least in our hands, CVs less than 5%, which is what we find is sufficient for a deep learning or a machine learning based platform. Now, the Diagnostic Cortex, we've developed this platform over the last 10 years to address a number of the problems with, clinical-based diagnostics. This is a completely custom algorithm and platform that, it's not an off-the-shelf platform like an elastic net or, any of the other tree-based methods. And what we're really trying to achieve here is to address a number of the questions that we run into and problems that we run into with clinical data sets, such as, when people think about machine learning and AI, they usually think about big data. where you're looking at, many thousands of, subjects or many thousands of instances of data that have either relatively shallow data or medium depth data, associated with each instance. In this case, what we're actually dealing with on the clinical side is we're dealing with, small numbers of subjects, on the order of a clinical study, anywhere from, 100 patients up to, 1,000 or 1,500 patients. And we're dealing with, because we're dealing in the omics realm, we're dealing with really large amounts of data. And that leads to the problem of overfitting, where you have many more pieces of information than you do subjects in the trial. You're really prone to, having so much data that for your given data set, you can find what's called the perfect fit, shown here by the red line, where you come up with a mathematical construct that perfectly fits the data. But what happens when you get this is that when you try to take it to another data set, that mathematical construct is so precise for your original data set that it doesn't recapitulate and it doesn't validate. So one of the things that we do with a machine learning-based overfitting algorithm is to look instead for the desired fit. So it's not absolutely perfect, but what it does is it goes and it finds that demarcation between your different outcomes, whether that be responsive to immunotherapy or non-responsive to immunotherapy, that you get a clinically meaningful differentiation between those groups, but not perfect. So you can see here that there is some overlap, between that green line, between the dark blue dots and the gray dots. And what that's allowed us to do is that allows our algorithm to achieve what's called generalizability, where, if we look at the analysis of this being like a lock and key model, if we're overfit, what we end up with is we end up with a single key that only unlocks our discovery set. So it's specific for that set. But by finding the desired fit, what we actually end up with is a master key that works for the discovery set. It works well, and then it works well in validation set after validation set after validation set. And that brings us to the question, of proteomics. So, one of the things that we use is we use mass spec-based proteomics, and that brings up the question of why measure proteins. And I think a lot of us have the view that, the canonical view of the DNA to the RNA to protein, where we can look at, changes in the DNA. We can look at genetics. We can look at transcriptomics, or we can look at simple proteomics and get an answer. But that's really confounded by what's really going on in this process. It's much more complex than that canonical, view of DNA to RNA protein than we think about. Here are just shown probably less than 30% of the biology that's actually going on to produce a protein, where we have, DNA, we have histone modifications that change, transcription of DNA to RNA. We have, methylation of the DNA itself at both enhancers and promoters that change not only the transcriptional level, but can change the isoform of transcription and the RNA that results. At the RNA level, we've got, a multitude of modifications to RNA that change its structure and change how it's transcribed to protein. And then we have whole classes of small RNAs like, link RNAs and microRNAs that actually block that translation to protein and, change the amount of protein. And then for protein itself, there's not just the protein, there are a number of different modifications that happen, things like phosphorylations, acetylation events, the addition of carbohydrates, the addition of lipids that really change the protein structure. And all of these play a role and can be used as biomarkers. But what the salient thing here is that what ends up happening is that the proteome really integrates all of these different changes into proteomics, and we can measure those with mass spec. Why do we measure the circulating proteome? Because what we see is that what's in the blood is very highly related to the hallmarks of cancer. So things like changes in metabolism, changes in the immune response, changes in mitogens and growth factors that lead to cell growth, changes in signal transduction, and changes in apoptosis. So it's a cogent place to look for in oncology, to look for biomarkers and predictive tests for what your cancer is doing. We have our deep Maldi mass spectrometry platform. This was what the company was actually founded on. It's a mass spec platform that really makes Maldi readily usable in the clinic. We get because of the algorithms that go into it and the way that we do it. What we're able to do is we're able to produce reproducible data that leads to clinical biomarkers. With this technique, we get finer mass resolution, we get much greater sensitivity. We're achieving on the order of six orders of magnitude of sensitivity in the circulating proteome. Most importantly, for clinical use, we get reproducible data. And with this technique, we're about 20 times faster than traditional Maldi. We've gone through, a number of generations of deep Maldi. Our original platform and our first generation on which we have, our VeriStrat product is based. With the Maldi spectra, and the computational alignment of those spectra and other things that we do with deep Maldi, we were able to reproducibly see about 100 proteins. We could definitely see many more proteins in the spectra, but we were able in this first generation to make about 100 of them reproducible. Our second generation that we've been using for the last couple of years, we were able to achieve the same thing with about 274 proteins in their translational modifications. This was due to, this increase was due to improvements in the methods used, the computational methods, as well as instrumentation. And now we're currently moving on to and completing our third generation of deep Maldi. This is based on a new platform from Bruker, where we can see 1,500 proteins. And we're currently moving on to our next generation deep Maldi platform, where we can see up to 9,000 proteins. And I think a good example of how this is used, we have, our what we call our primary immune response classifier in our pipeline, where, we can predict, who's going to respond to immunotherapy or not. But I think a really good example of that is some work that we did with Genentech for their asset atezolizumab, where we started with 77 non-small cell lung cancer patients and developed a test that predicted those patients that would respond versus those that wouldn't. At that point, we locked the algorithm, and we took that to a blinded independent validation, where here we were able to recapitulate that data, looking at the solid lines here, where we see a large difference between the responders to atezolizumab versus those that don't. And we were able to show in this study as well that that was predictive for atezolizumab because it didn't split any chemotherapy arms. So here we're looking at docetaxel, where it didn't do anything prognostic or predictive for docetaxel, nor any of the other chemotherapies that we tested. And the last point is that one of the questions that we commonly get with this technique is, how do we know that we're not just measuring something like mutational load or PD-1 expression? And what we were able to show in this study as well is that that this technique is really orthogonal to other biomarkers for for immunotherapies that are out there, like TMB, where TMB didn't really show a split here. Our classifier did. And with that, I will wrap up and take questions.